Hello everybody, thank you for tuning in again. Today we're going to talk about DNA replication. As you can see, we have a guest star here who's going to help us go through this. So, starting with DNA replication part 1. Part 2 is going to be about bacteria. Part 1, we're just going to talk about it, how it would happen within us. So, when we look at DNA replication, basically what's happening is just DNA is being copied. Again, keep in mind back when we talked about mitosis, we went through all those different stages, and in interphase, in that S phase, the synthesis, is when it was being replicated. That is what we're going to talk about specifically. Keep in mind how important that is, because we don't want to have any errors or mistakes in that copying. And so when cells are created, each new cell has to have its own DNA to function properly. Because again, that DNA holds all the codes, that gives it its proteins and all the other different aspects of it. Lives inside the nucleus, which is our control center of our cell. So obviously we need that DNA in each of our cells. When the DNA replication occurs, it occurs prior to mitosis. Meiosis actually taking place. Again, it happens in that S phase. So just go through that cell cycle again. We have interphase, which is G1, which is growth. S, synthesis, copying the DNA, which is what we're talking about. G2, second little bit of growth, and proofreading that DNA replication. And then we go into prophase, metaphase, anaphase, telophase, and then finally cytokinesis, where the cell is actually splitting. So, in DNA replication, there's going to be two types of enzymes that we're going to talk about specifically. Now, keep in mind an enzyme is a type of protein. We talked about that a little bit ago. Proteins are our most diverse macromolecule. For the reason being is they can be so many different things, one of which is enzymes. And again, keep in mind, we always know that things that end in ACE are enzymes. So helicase and DNA polymerase, both enzymes. They have both separate jobs, but both equally as important for DNA replication. First thing is helicase, which is going to unzip and unwind the DNA molecule. Because again, our DNA looks like this up top here. So this twisted ladder, you could say. In order to replicate it, we're going to have to separate that, and we're going to have to straighten it out. So the helicase comes in and unzips and unwinds the DNA. And so that enzyme is going to break those hydrogen bonds. So these bonds right in between, if you look over here to the right, between this T and A, that's a hydrogen bond. Again, those are weaker. So it's going to break that bond, which is going to allow it to separate the two sides. Again, this sugar phosphate backbone here, we don't want to break that. That's held together by covalent bonds. We do not want to break that. Think about that as like your backbone. You don't want to break your back. That would definitely not be ideal. So the helicase comes in, unzips, unwinds the DNA molecule, and then the replication fork is right here. It's where it's separating. So that helicase just binds in here and is traveling along this DNA, unzipping and winding it as it goes. Now, what you'll notice in the diagram over here is after it's unzipping and winding it, we're getting a new strand being created on both sides. That's going to be very important because, again, this one will go to the new cell. This one will go to the old cell, or stay in the old cell, you could say, as it's replicating. This is actually a very important topic with semi-conservative replication as well. So semi being half, conservative save, replication obviously being copy. We're using our parent strand as a template. That's going to help limit errors and mistakes while copying it. So each new cell still has an original copy of the DNA and then a strand that's new. So it's an old strand with a new strand, old strand with a new strand in each of the cells after replication has taken place. So in our diagram here, you'll see the blue strand is the original DNA. So a parent strand, you can follow that all the way down on both sides. And then you can see that blue strand is actually being read. So notice we have A, G, T, G, C, T here. So then this new strand here, this yellow strand, is going to have that complementary base pairs. When I say complementary, I just mean if there's a T, it brings an A. If there's a G, it brings a C. If there's a C, it brings a G. And if there's an A, it brings a T. Right? Riley approves. Yes, so those nucleotides are brought in by DNA polymerase, and polymerase is actually going to be the one that's going to be traveling along, reading it, and then bringing in those complementary base pairs. So our helicase unzips and unwinds the DNA, and then our polymerase reads it and brings in the complementary base pairs, complementary nucleotides for it. And then that yellow strand is our new one. Okay? And again, complementary just means it's matching pair, you could say. So one thing that's kind of neat about it, these new strands are opposite directions. So basically what's happening is if we start up here and go this direction, it's the same as down here to that direction, basically. So they go opposite directions. However, they do have these same pairs because, again, they're both the same halves. It's just kind of reversed. It's like two sides of the highway, driving down the highway. Still the same highway, just going opposite directions. Now, in DNA replication, one of the new strands is created from the bottom of the blue strand, working the way to the top, and then the other one is working from the top down to the bottom in small pieces. So the reason that it's doing that is because our polymerase can only really go one direction. So it does the other one in fragments, and I'm going to create a video kind of drawing and showing this, so that'll make a little bit more sense. But those fragments are known as Okazaki fragments, named after the Japanese scientist who discovered them. But that piece that's done in chunks or in fragments is going to be known as the lagging strand. So there's the leading, which is just a nice, smooth, continual replication. And then there's the lagging one, which goes in choppy motions because that polymerase can only travel in that one direction, which again will make so much more sense when you watch the other video with me drawing it out for you. So our DNA replication, our end result is two identical DNA molecules. Before I continue going back to this up here, not too concerned about you fully understanding all this. Ligase is just one that's going to connect all those fragments. Helicase and polymerase are going to be the main ones, but talking about ligase kind of shows you what's going on a little bit more in depth, just as a heads up there. So anyway, back to DNA replication. End result is two identical DNA molecules. Half of each are going to be the old one, and then the other half is going to be the new. Again, that's known as the semi-conservative replication because one strand is new, one was used as a template to create the new one. Again, limits mistakes. We don't want mistakes in our DNA. That causes mutations. Mutations could be good, bad, or indifferent, but I'd rather not test the waters in having mutated DNA. And then another diagram that kind of shows you what's going on with the leading and lagging. So we have our DNA up here. So here's our replication fork where it starts to split. Our helicase is here, traveling up along it, separating it. Our polymerase is going to be here, going continually through on this side, and then here doing it in short fragments as it opens up. So this polymerase here will just be following right behind the helicase, adding those new complementary nucleotides, whereas over here, notice it does it in little fragments. So as this opens up again, a ligase will come in here, or a polymerase will come in here and do a little more section. And then as it opens up here, polymerase will come in, add a couple more complementary. This would be your lagging strand. This would be your leading strand. Again, it'll make a little bit more sense after I draw mine and kind of explain that out a little bit more. I'm going to try and use a whiteboard app. We'll see how it goes. But that is all for DNA replication. So if you have any questions, please reach out via email, and I'll get back to you as soon as possible. Thanks for watching. Say bye. Bye. Bye.